Erev Tov Herzliya. Good morning, Cleveland, and greetings to our friends around the world who are calling in today for the America-Israel Friendship League webinar series. I'm Wayne Firestone. I'm the Executive Director of the AIFL in the United States, and we're excited to continue this series that we started now with the Zuckerman Institute about high science issues and amazing collaborations that are going on around the world, brought together by this amazing Zuckerman Institute, which has focused on postdocs, has focused on what the next generation of innovation and research will be. We had an exciting kickoff to this a couple of months ago with a segment on longevity, and we're continuing the series today and really excited to bring you two world-class scientists and researchers in collaboration. I'm thrilled to have as a colleague, Lena DeShilton in Tel Aviv, who is the executive director of the Zuckerman Institute. She has a background in development and marketing. She's founded a company that actually allowed for international science and medical communities to collaborate and come together at international conferences. Previously, she was worked as a director of marketing at the Ministry of Tourism for Israel. And so she really brings together these two worlds that we've seen in this period of COVID, needing to find new ways to be able to communicate and collaborate. So Lena, thanks for being our partner on this. And I turn over you to introduce this next segment in our series.
Thank you, Wayne. Good afternoon and good evening to all of us and to our friends in the US, Israel, and wherever you are connecting with us. My name is Lena DeShilton, and I'm the Executive Director of the Zuckerman Institute in Israel. Today, I'm honored and very happy to introduce you to our second webinar presented in cooperation with the America-Israel Friendship League. The Zuckerman Institute was established in 2015 to achieve the philanthropic vision of Mortimer Zuckerman, to enhance collaborations and communications among the most promising scholars in science, technology, engineering, and math in Israel, the United States, and Canada. The Zuckerman STEM Leadership Faculty Program provides vital resources to Israeli universities, allowing them to compete with top North American institutions for the most promising candidates. The program facilitates the return of top Israeli scholars to Israeli institutions, cultivates world-class scientific talented, and in return attracts outstanding postdoctoral researchers from top US universities, thus creating a cycle of excellence. Faculty scholarships are given out to every year to each of the 7 participating universities, including Bar-Ilan University, Ben-Gurion University of the Negev, Haifa University, Hebrew University of Jerusalem, Technion Israel Institute of Technology, Tel Aviv University, and the Weizmann Institute of Science. Since the start of the program in 2016, 30 Zuckerman labs have been established in Israel. We are extremely proud of that accomplishment. On today's web, we have 2 presenters, a Zuckerman faculty scholar from Haifa University in Israel, Dr. Shani Stern, and our Canadian collaborator and colleague, Dr. Martin Alda from Dalhousie University in Canada. Now, I'm happy to welcome and introduce and put the mic back to Wayne, Executive Director of the America-Israel Friendship in the US. Wayne, the floor is yours.
Thanks, Lena. This is such an amazing opportunity to bring together and share in this larger global platform what a scientific collaboration really looks like, which is at the very heart of the Zuckerman Institute activities. Dr. Shani Stern has this fascinating background. She starts her undergraduate at Tel Aviv University as an electrical engineer and begins to work in the world-renowned startup nation high-tech industry. And then she goes on to, in her graduate works, get a master's in computer science and a PhD in physics from Weizmann. She continues to do research that ends up taking her to San Diego with her family, and begins to collaborate with 3 different psychiatrists around the world about different biological processes, ultimately that will impact on her specific field of study in bipolar disorders. So with that, as sort of a general thumbnail of her background, let me introduce one of those 3 psychiatrists who she's worked with, Dr. Martin Alda. He is a professor of psychiatry at Dalhousie University in Halifax, Canada, where we have many people calling in from Winnipeg and Toronto and Ontario. So many landsmen that are indeed interested in your work that are with us on the webinar today. He's originally born in the Czech Republic and studied psychiatry at Charles University in Prague. He is a clinician scientist who pioneered molecular genetic studies using lithium treatment for bipolar disorders, even at a time where that was not necessarily the most popular treatment. He's received numerous international prizes for brain research. And really, I think this collaboration will give us a peek behind the curtain of what it looks like to really have scientists in different disciplines and in different parts of the world working together. So don't get frightened by any of their slides. They have promised to share with us and answer any questions for those of us who are not academics and have PhDs in the sciences. The intent of this series really is to actually share some of the groundbreaking research and make it more accessible to the general public, so we can all be watching the science headlines on the one hand, and on the other hand, really begin to demysticize some of the important breakthroughs that are happening very much during this important period. So Dr. Alda, let me start with you to sort of frame more broadly the fields of mood disorders that you're working with, and then we'll add that as a complement to Dr. Stern's work, specifically focusing in on bipolar disorders.
Okay, so thank you. Greetings, good morning, good afternoon, good evening from Halifax in Canada. And in the next 10 to 15 minutes, I would like to take you through some of the dilemmas that we face when we both care for people with bipolar disorder and when we do research. I thought that the best introduction to that might be if I present a clinical case. So let's call this person AB, and I need to stress that all the details that would allow identification of the person have been heavily modified. So this is an anonymized case history. So the AB we saw when she was 49 years old, when we were doing a family study of bipolar disorder. And she was a wife and mother of 4 children. And her history was as follows. When she was 16, she had an episode of depression that took about a few weeks. She was prescribed something she didn't remember. And she took it for several months and then forgot about the whole thing. And then when she was in her 20s, she had many episodes, usually a week or so at a time, when her mood would be up, she would be very busy, didn't need to sleep. She would be up in the middle of the night, baking, cleaning the house, going from one thing to another. She would speak very fast, her thoughts would be racing, and then things would settle completely again. And then after giving a birth to one of her children when she was 28, she had really severe depression that took about 2 months, and then she recovered again. And 3 years later, she had another severe postpartum depression. And at that time, she saw a doctor and was prescribed lithium. Within 2 weeks, she recovered. And then for the next over 20 years, she just continued the lithium at the usual dose, no other treatment, and was completely well. And then several years later, after we saw her, several years after the assessment for the genetic study, we learned that her lithium was discontinued by her family physician, and it was replaced with another medication because there was concern about possible side effect from lithium. And then a year later, she died by suicide. Now, when you look at the family history of AB, there are some clues that should alert you to this risk. Her uncle and her aunt also died by suicide, and she had 2 sisters with bipolar disorder. And so the history reminds us that the kind of old aphorism by William Osler, medicine is a science of uncertainty and an art of probability. So how do we tackle this uncertainty and probability, both clinically and research, and how the two can inform each other? Well, bipolar disorder is a severe illness. It is characterized by episodes of mania and depression, as we saw with this lady AB. She often hits people at a young age and then runs a lifelong course. It is familial and it is recurrent. Without treatment, the depressions and manias keep coming back. It is also a lethal illness. The mortality of bipolar disorder is almost 3 times that of the general population. But importantly, it can be treated. And here I wanted to show you data from one of our studies several years ago, where we looked at the risk of suicide among people with bipolar disorder. And as you can see, the risk of suicide is especially high in the first 5 years of the illness, but it never goes down to 0. It continues throughout the lifespan. And how do we do compared to other fields of medicine? I borrowed this slide from Tom Insel and modified it, and wanted to show what happened with some major causes of mortality over the last 30 to 50 years. When you look at stroke, the number of deaths in North America have decreased by at least 20,000. In case of HIV/AIDS, the lives saved over that period of time have been about 30,000. Very significant drop in mortality from heart disease. And in case of childhood acute leukemia, the number of deaths dropped by about 6,000 over that period of time, but it was also an 85% reduction in overall mortality. And compared to that, psychiatry has not done too well. The deaths from suicide remain about the same. And the factors that probably are responsible for these medical successes are early diagnosis and intervention. For instance, if you have a stroke and can get to the hospital within minutes to few hours, your chances of survival are very good. Knowledge of disease mechanisms, that was especially the case with the HIV and AIDS, and also personalized treatment, and that's what we are going to discuss today. In leukemia, for instance, the therapeutic spectrum has not expanded too much, but hematology is much better in matching the right treatment to the right person. So how do we do in psychiatry with bipolar disorder? Well, it takes usually several years to get to the correct diagnosis. The illness often starts with depression and takes a while to diagnose actually that the depression is in fact bipolar disorder. And then people take treatment from a long list of first and second line options, and you have to usually wait several months to see whether the treatment is working or not. The Canadian guidelines actually list 9 first line and 7 second line options. And in the meantime, during these trials, people can experience suicide, disruptions in jobs, schooling, family life. And what we would like to do is we would like to be able to come up with genetic or molecular markers that would tell us that this type of illness will be treated best with this type of medication. And that takes us to lithium. This is from the Atacama Desert in northern Chile, where much of world's lithium comes from. And some of the earliest studies of lithium prevention of manic and depressive episodes come from more than 50 years ago. Here we have data from a classical paper by Mogen Sko, that's the person on the right-hand side of the photograph, together with Paul Groff and Bruno Miller-Erlinghausen. Oops. And as you can see, there are people who would have multiple episodes of depression, like here, and then when they get treated with lithium, the episodes disappear here or here. But also, there would be people like this one who might have some episodes before lithium, and the medication doesn't help at all, or in this case, the same story. The patient, in spite of the treatment, continues experiencing either depressive or manic episodes. And so the question is, how do we differentiate? How can we tell who is a good candidate for lithium and who is not? And we have some data from decades of studies showing that maybe bipolar disorder responsive to lithium is a specific form of the illness. Here we have data from a 20-year follow-up of people with bipolar disorder treated with lithium, done by an international consortium of which we are part. The gray bars are the numbers of people followed for that length of time, and the red line would indicate the overall morbidity over the years. So what you can see is that those people who are well in the first few years of treatment usually stay well for many, many years after, up to 20 years of follow-up. And we also did a positron emission tomography study in which we compared people who responded to lithium and people who responded to another mood stabilizing treatment, valproate, and saw a major difference in activation of very important brain region that is involved in mood regulation, the so-called anterior cingulate area 24A. So that tells us that lithium response is rather specific and unique to a group of people with bipolar disorder. And what we also clinically saw and what we have been studying for the most of the last 20 plus years is that the treatment response also runs in families. Take a look here at pedigree of people with bipolar disorder, where 3 people who were treated with lithium all did well on it, and another family like that. And I could show you dozens of families where we have similar data. And then there would be families like this one, where out of 3 people treated with lithium, none responded. So it seems like the response itself runs in families, not only the illness, but the response to treatment. And when we analyze the data together, we find that in people who have a close relative who has done well on lithium, you have odds of almost 8 times to do also well on lithium, and almost 5 times better chance of responding to lithium compared to somebody where you don't have any information, where we don't have family history details. So this is an important observation that led us to analyze such molecular data in families, and I think Shani will tell you more about the exciting work with the molecular markers. I would just like to close, go back to the AB case that we started with, and we asked ourselves the question: if you have somebody who has done well on lithium, how likely is that person to do well on other treatments? And so we used machine learning methods where we tested response in over 1,300, almost 1,400 patients evaluated for lithium response, and almost 300 people treated with a different class of medication, so-called anticonvulsants. And we saw that if we trained the model on response to lithium, we could get 80% plus accuracy predicting who will do well, while that model could predict response to the anticonvulsants with lesser accuracy. And there are clinical features that differentiated the responders to lithium and the anticonvulsants. Importantly, when you think about the AB case, lithium response has no bearing in relation to the association with suicide behavior. So the fact that the patient is at risk of suicide doesn't tell you whether lithium will be effective or not. So you might have a good chance of getting good effect with lithium. With the anticonvulsants, the responders typically had no suicidal behavior in their histories. So you can see that the case that I presented would suggest that that lady should not have gotten the anticonvulsant medication, and it might have actually protected her. So I'll stop at this point and hand over to Shani, and she will have the more exciting part of the presentation. Thank you.
So thank you, thank you, Martin, and good evening to everyone from Haifa. I'm starting to go into the. So I'm going to tell you about work that I did, a lot of it in the Salk Institute during my postdoctoral training, and I'm continuing here in the University of Haifa. And I was very lucky to collaborate with Martin on this work. We got really great data, and we shared many great data. So over the last few decades, many brain disorders and diseases were modeled using animal models. The problem is that when we're talking about neuropsychiatric disorders, usually the genetics is not well defined. It's very hard to get the same genetic changes in a mouse or rat model. So in a rat or mouse model, we usually take the genetic change that we think that there are and make it in the model, and then we have a good model. The advantage of animal models is that the neurons that we're going to do the experiments with have developed over in a brain, in an animal brain. They received all that they needed for correct differentiation and maturation. So thanks to the pioneering work of Professor Shinya Yamanaka from 2007, today we're able to take adult cells, such as we see here fibroblasts or PBMCs. These are parts of the blood. And we're able to cause them to reprogram them and to cause them to kind of go back in time and become stem cells. So we can see here, this is the schematics of the process. We infect the cells with some virus that contains 4 transcription factors, 4 proteins that causes the cells to overexpress them. And we can see here, this is a transformation that we made in my lab in Haifa. We made these started as PBMCs, and after a few weeks, they became stem cells. So thanks to this method, now we have stem cells that we can derive from the patients. Now we can differentiate them as neurons. The neurons that we derive eventually have the exact same genetics as the neurons in the patient's brain. So this is a very good and precise model. So all that we do not even need to figure out what the genetic changes are in the patient, and psychiatric patients often they don't have a specific genetic change. They have very subtle, small changes that over many small changes may accumulate to some disorder. So we don't have to figure it out. We have now we have neurons with the same genetics. The disadvantage is that these neurons, they matured in a culture dish. So of course we try to mimic the brain environment, but we are not. We don't have the exact recipe. So when today we are also able to decide which type of neurons we want to differentiate the cells to. So we can be quite specific. When we started this project, we decided to go with the hippocampus, which is an area in the brain that is known to be affected in a bipolar disorder. So these are some, just very few, but of course there are many studies that have been performed showing different activation in the bipolar disorder or parts of the hippocampus. Changes to the volume of the hippocampus, some changes to parts of the specific types of memory. So we can see here, this is the human hippocampus. This is a mouse hippocampus. We went specifically to differentiate a type of cell that is in the dentate gyrus. This is a part of the hippocampus. It's called the dentate gyrus granule neurons. And the dentate gyrus is a special area in the brain. It's one of the only places that adult neurogenesis occurs. So, that neurons do not divide throughout their lifetime. So, but so we don't have any new neurons forming throughout our lifetime, but in very specific areas in the brain, very few, there does occur adult neurogenesis, so new neurons, a few hundred neurons are born every day. And the dentate gyrus is also responsible for helping in pattern separation when we see images that are close by and we try to distinguish them, and has functions in learning and memory. So the cohort that we received from Martin, it consisted of 4 healthy individuals that did not have the disorder. And then we had 6 bipolar disorder patients. 3 of them were lithium responsive. So we can see here that when they were off lithium, they had mania and depression episodes, but with lithium they did not have it. And we had lithium non-responder patients, so they continued to have episodes after the lithium treatment. And I will mark this as LR and NR for lithium responsive and non-responsive. So we started by making iPSCs, induced pluripotent stem cells. So we can see, these are some, we have a way to see what type of proteins are expressed in the culture dish. We have antibodies, so the antibodies bind to specific proteins, and they have some fluorophores that we can image. So we can see here, these are the iPSCs that were made from all the patients and the control individuals. And we can see, that they express many of the pluripotency markers. And we also performed karyotyping, which came out correctly. And next, we differentiated them using a protocol that was developed at the Gage Lab, where I did my postdoctoral training, to make dentate gyrus granule neurons. So this is basically the protocol. During the protocol, we also make neural progenitor cells. So these are still dividing cells, but they know that they're going to be either neurons or astrocytes. And we see that the control, the LR and the NR, have similar levels of these markers for neural progenitor cells. And finally, after approximately 6 to 8 weeks, we have neurons. We can see this is the staining of the neurons. And we are looking for a specific protein, which is called PROX1, which marks the dentate gyrus, specific for the dentate gyrus granule neurons. So overall, if you can see the 3 groups, the control, LR, and NR, they all have approximately 50% of the neurons in the dish are PROX1 positive. And we have a marker for them, so we can do the experiments with these cells. So I started with an experiment which is called the cell patch, patch clamp, which I'll explain in 2 minutes about it. But generally, it allows us to see activity from the neurons. So what is this activity? These are action potentials. We can see here how they look. They look like spikes. Sometimes we call them spikes. And these are abrupt changes to the membrane potential of the neuron. And what was interesting was that if you can look at the LR and NR recordings, the bipolar neurons have more of these action potentials, so they are hyperexcitable. So this is one example, but over recordings over hundreds of neurons, we see the statistics is very clear. Both the LR and NR groups have more action potentials, so they are hyperexcitable. So what is this patch clamp? So if we're talking about a neuron, so this is a schematic or a cartoon of a neuron. This is like a part of the neuron. We can see here the membrane. This is the inside of the neuron. This is the outside of the neuron. Inside the neuron, we have a high concentration of potassium ions, and outside we have high concentration of sodium ions. And on the membrane, we have these pores that can open and close according to environmental conditions. So when the neuron is at resting state, these pores are more open to potassium ions. So by diffusion, potassium bypasses the membrane and starts to diffuse over, but this creates a gradual electric charge to build in the inside of the neuron on the membrane because these are positive ions, and this causes an electrical force. So when the electrical force equilibrates with the chemical force, this process stops. This is the resting state of the neuron. It occurs when the membrane potential is approximately minus 60 millivolts. So just to compare, over the socket, we have 110 volts. So this is about a 1,000th, a little less. So it's small potentials. So, and whole cell patch clamp was invented in the late 70s by Erwin Nair and Bart Zuckerman. They received a Nobel Prize for this. And it allows us to see these potentials and currents, on specific currents in single neurons. So what we do, we have a pipette with an electrode, and we touch the cell. By applying a small suction, we fuse with the cytoplasm, and we are able to record, and then we have an amplifier and digital analog to digitizer, which we can go record the signals in the computer, and we are able to measure these potentials and currents from the cell. This is the action potential. In the action potential, we start at the resting membrane potential, which is minus 60 millivolts. Then due to some change in the environment, usually it's because other neurons fire and they are connected with our neuron, there will be a small change to the membrane potential. This causes the sodium ion channels, which are highly sensitive to these changes in potential, to open up. Then sodium rushes into the cell by diffusion, and this causes the membrane potential to depolarize very abruptly. So now we have a membrane potential changing from minus 60 millivolts to approximately 20 millivolts. And at that time, the potassium channels open and sodium channels close, and then potassium goes out of the cell, hyperpolarizing the membrane potential back. After, in the end, it reaches a steady state. Before the steady state, there is a small after hyperpolarization, kind of an overshoot. So we can see here an example recordings where we hold the cell at a specific potential, and then we record the currents. So a downward deflection are the sodium currents, the upward deflections are the potassium currents. So as I mentioned, we saw that the bipolar neurons were hyperexcitable. But one of the things that I noticed was that despite the fact that they were both hyperexcitable, actually the physiology was very different between the lithium responders and the non-responders. So we can see here in the example that the lithium responder has a high amplitude and a narrow spike, while the non-responders have a low amplitude and a wide spike. And this prompted me to think whether I can define maybe some features and use them to try to predict which of the patients would respond to lithium treatment. So for example, I defined the spike height, which is where the threshold for the evoking the action potential until the maximum, that's the spike height, or the spike width, which is, we measure it half the amplitude. And so I had approximately 8 features. And then what I did is I built a computational model that takes 5 out of the 6 patients, study their, or trains the model on their electrophysiological features, and then predicts the electrophysiological features of the 6th patient. And we do this iteratively. Each time we take a different patient out and we train the model by the other 5. And in the end, we pull together all the predictions to assess the performance. And we were able to receive this way a predictor that gave a very low error rate. So we're talking about a few 2 to 5% error rate. So next, we wanted to see whether if we take a different type of neuron, we would also see these changes. So at that time, we developed a protocol for making CA3 pyramidal neurons. So CA3 is another area in the hippocampus. So just by changing concentration of a protein that is expressed during the development, we were able to get CA3 pyramidal neurons. So then I differentiated the cohort again as CA3 pyramidal neurons. And as CA3 pyramidal neurons, what we saw was that only the lithium responders were hyperexcitable compared to the control. The non-responders were not hyperexcitable. So how does this hyperexcitability look like? So we can see here an example of a lithium responder neuron recording versus control in NR. So the hyperexcitability occurs in the high current injection. So when we inject with a high amplitude current, the control and the NR neurons cannot sustain the activity, while the LR neuron can sustain the activity, keeps on firing, kind of like super neurons. So we were trying to find a mechanism for why is this symptom that we see of the phenotype of the hyperexcitability. So one of the things that we measured to be different was the fast potassium currents. So the fast potassium currents occur right, this is the fast potassium current right here, right immediately after when we give a depolarization step of the potential of the membrane. And we can see that the fast potassium currents were higher amplitude, both in CA3 and in DG neurons for both the lithium responders and non-responders. So we can see here an example. We can see also that the lithium responders, for example, had a very fast kinetics, which pointed us to specific types of these fast potassium currents channels. Another current that was highly implicated was the sodium current. So the sodium currents were very much reduced both in DG and CA3 neurons in the non-responsive bipolar patients. So we wanted to see how does this affect the excitability. So we plotted the correlations. So the sodium current was, the excitability was correlated with the sodium currents in all the three groups, which is something that we know from the literature. This is a neuron which has higher sodium currents is more excitable. But what was surprising was that the excitability correlated with the fast potassium currents in both the LR and NR groups, which is surprising because it doesn't happen in the control neurons. And so also something that we know from the literature, it's not supposed to correlate positively at least. So we thought, okay, if there's an implication of the fast potassium currents, let's try to block them. And by blocking them by 3 different types of potassium channel blockers, we reduced the hyperexcitability significantly in the lithium responders. So another phenotype that we noticed was we termed it physiological instability. What we saw was that the control neurons, they tended to be somewhat, the diversity between them was not very large. If we look, this is an example of one-day recording or one experiment. Each of these graphs presents one neuron. And we can see that if we look between the neurons and if we go ahead and count the action potentials, it would not be very different from cell to cell. There is, of course, differences, but it would not be so strong. But if we look at the NR neurons, for example, we can see most of the times we would have something that looks like this. Some of the cells would be very hyperexcitable, like this one, or this one, or this one. But then some cells would be very hypoexcitable, like producing almost no action potentials, like this one or this one. And this happened approximately in 70% of the times, but approximately 15% when doing many experiments. In approximately 15% of the time, we would have what we termed a global hypoexcitable state or day, where all the neurons did not evoke any action potentials. And this kind of reminded us a possible correlation with the depression state. And approximately in 15% of the times, most of the cells, these are different times, but would be hyperexcitable. So, I come in one day and almost all the cells that I record in the NR neurons are hyperexcitable. This kind of reminded us over a possible mania state. So when we applied lithium, we treated the cells with lithium for 2 weeks. And then we saw what happened. And indeed, the lithium reduced the hyperexcitability in the lithium responder neurons. And another phenomena that we saw was that when we applied lithium, during the development, the cells grow. And both the LR and the NR cells, the bipolar cells, grow faster and they're larger. So lithium reduced this kind of overgrowth and made them more similar to the control. So also the size of the cells it did reduce. So it actually didn't reduce it, it grew slower, more like the controls. So, and then we did a completely different type of neuron, motor neurons, which we didn't think would be implicated. And for these, just to see the specificity. And indeed, for motor neurons, we could not find any significant changes in the electrophysiological features. And finally, this is work that we started now with Martin regarding biomarkers. So we're interested to develop the classification or the prediction to something that is more usable in the clinic, in the sense that it would be much faster and cheaper. So what we did is we sequenced, we performed RNA sequencing just to measure the amounts of RNA in the lymphoblasts themselves. So right now we're not differentiating, we're just taking the lymphoblast, which is a small change from the lymphocytes, which can be acquired from the blood cells. And we saw many genes that are differentially expressed, and these are the most strongest differentially expressed genes. It was interesting to notice that the SCN3A, which is a sodium channel gene, was differentially expressed. And just to remind you, this is the way that the electrophysiological recordings look like. We see the sodium channels are very much reduced. And this is also RNA sequencing that we did in the neurons themselves. So we can see several types of sodium channels that are very much reduced in expression in the NR neurons. So we think that sodium channels may have some strong implication in lithium response. So, and this is all the people that helped me with this work. Thank you all.
Well,
Now-
that particular slide is one of the ones that I actually find the most fascinating, and it's one of the things that I wanted to explore a little bit with Dr. Alda, and how he connected with you initially in this work. Dr. Alda, the slide that you showed in your presentation comparing the mortality rates of other known disease, I thought was really interesting. And I guess the implication was you're not satisfied with where the current state of treatment is. And I know that's one of the things that has excited you about this breakthrough in the work with the STEM work in neurons. Can you, at what point did that, did you realize you were going to need to find and collaborate with someone sort of and take some risk and innovate in a new area that didn't currently exist? Can you take us to that part of the story?
Yeah, that's a great question. Well, you know, what I was showing in my presentation was that a lot of treatment in psychiatry is essentially trial and error. And we have been interested to take that element out of clinical decisions. So we started with studies looking at molecular genetics for which you need large samples. But the difficulty in psychiatry is that you know, a lot of work that we do is very indirect. Compared to other areas of medicine, we don't have direct access to brain tissue for obvious reasons. And so when the stem cell technologies appeared on the scene, that really I felt was a game changer. And so we connected with several other research groups and collaborated. And that took us to collaboration with the Salk Institute, where Shani was at the time a postdoc, and she did that great work characterizing the physiology of these neurons from responders and non-responders to lithium that showed such clear-cut differences. And I also wanted to stress that the work can be not only helpful in differentiating who responds to lithium, who does not, but also can serve as a platform for development of novel treatments and screening treatments. There are thousands and thousands of molecules that have been already tested in different conditions. We know that they are clinically safe, but nobody thought about using them in people with bipolar disorder. And if you took that trial and error approach, it would take centuries to try all these medications. Now you have a model of the illness essentially in a laboratory dish. So it offers you a significant shortcut to testing whether something has the potential in reducing that hyperexcitability and thus treating bipolar disorder.
You know, Dr. Stern, as I recall, you originally did a lot of your research with animals, with mice, and then at some point you have found this new path. Can you just describe the time period in which, you know, all that work that you showed of your trial and error and working with neurons from people who would use the lithium treatment and not? Can you describe to Dr. Alda's point, like how fast did you accelerate this process through this advanced technology you're using now?
So you mean that, like, the process of how fast it can accelerate the process of finding like which of the patients would respond?
Yeah.
So yeah, so I think Martin knows better than me, but I think some of the patients take months and even years, right, to diagnose. And the method that I did with the stem cells, with the neurons derived from stem cells, this is also a relatively long. So it, from lymphoblasts to iPSC is usually approximately 4 to 6 weeks, and then to neurons is maybe 10 more weeks. But it's also a costly method, and also you need someone to do patch clamp, which needs some specific technique. But that's why we're now working to find something that would be much faster. And we have also right now some exciting preliminary results, but we're continuing this. We're hoping to find something that you would be able to come to the clinic and within a week get a response, like you have a 95% chance to respond well to lithium, or and then we can expand it, as Martin said, to other treatment.
Well, again, one of the interesting points you made, Dr. Alda, in your slide is this idea of personalized treatment. And we had a question come in from one of the viewers. Do you see modern psychiatry moving toward more customized medication, similar to what we're seeing with modern cancer treatments?
I hope so. That's been certainly our effort, and where we have invested a lot of our research and time. Following also on some of the previous comments, there are data suggesting that an average person with bipolar disorder takes about 10 years from first manifestation of the illness to effective stabilization. We have been looking at mainly the treatment selection, but the stem cell approach could also help in differentiating, for instance, who is developing bipolar disorder. If you have somebody with major depression, for instance, and you find that the cells are actually hyperexcitable, as Shani was showing, that might lead you to suggest possibility of bipolar disorder. So we are also looking at whether the risk of suicide could be uncovered through these studies. So it's not just the treatment selection, but as a whole, it would be a package of kind of personalized, individualized treatment of bipolar disorder.
Yeah, I found that fascinating that when you showed the family tree, that the implication for this is both on the diagnostic side in terms of being able to identify people that may have the condition in their family, as well as on the treatment side in terms of seeing where you may have a better likelihood of success with lithium. Maybe there's a question that came in from Diane, just trying to understand the scope of the bipolar disorder in the general population. Do we have a sense of how many people actually in the general population carry the gene? And there was a question of whether in the Ashkenazic Jewish population, there is a recognizable higher percentage. She had seen a number of 40%. I don't know if that is accurate or not, if either of you would know.
The prevalence would be about 1.5–2% in the general population of people who have bipolar disorder. The difficulty to tell that somebody has or doesn't have bipolar gene is that we have access to data from tens of thousands of people, but when you lump all different forms of bipolar disorder together, it's more difficult to tell whether actually the illness is associated with one specific gene or multiple genes. Our recent study of 40,000 cases of bipolar disorder, kind of genetic mapping, identified over 100 possible genes. The question is, do you need all these 100 genes in conjunction, in combination, or is it simply that there are some families or some people where it's one gene of those 100, and another patient or another patient has a different gene, and that has not been resolved?
Dr. Stern, I want to, when we spoke earlier, so much of your work now and so much of this progress is based on the use of the STEM. At one point that was controversial, but I take it that that's no longer a barrier in terms of any of the work that you're doing.
Yeah, so we're using the induced pluripotent stem cells. So these are not embryonic stem cells. So we don't use any fetuses. We start with a simple, usually simple blood draw. And then if you remember from my first or second slide, we make them into induced pluripotent stem cells. So we have no problems. Of course, we go through all the ethical procedures and we have ethics reviewed, and it's very easy to get approved because all we take is a simple blood draw.
So one of the questions that came in from Moti for you, Dr. Stern, relates to a different, perhaps controversial new technology, CRISPR. If indeed you're able to identify now the defective genes, Moti is asking, why wouldn't you just cut out the faulty gene? Is that an option?
Yeah. So first, there's no one gene, but there are, like, for example, we just recently published also something showing that LEF1 gene is so the non-responder patients have LEF1 gene that is very much reduced in expression. So we can target specific genes through experiments of such as RNA sequencing and so forth. And definitely this is something that it will be something it's not very far. So I think that gene therapy is now starting with the patients that have, like, we have patients that have very strong, not bipolar disorder, but patients of other symptoms or other disorders that die very young. So this is now something that is starting, and at some point, I'm sure it's not very far, when the technique will be safe enough, it may be used for such patients. Yeah.
Dr. Alda, I guess one of the implications of actually knowing from Dr. Stern's work whether the lithium treatment is likely to be more likely to be successful and desirable to use is if you find out it's not going to be, and you do know that you have to look for alternatives. One of our viewers is asking about medical cannabis. Do you find that this is one of the options, or is that sort of being viewed as a more too wide a solution based on the data today?
That's a complicated issue, partly because, of course, there are lots of controversies and uncertainties about cannabis. And for instance, there are some suggestions that cannabidiol, not the tetrahydrocannabinol, but cannabidiol, might be effective in acute bipolar depression, but that's just very preliminary data, and we are still waiting for the more kind of conclusive trial. At the same time, the THC, tetrahydrocannabinol, is known to increase the risk of psychosis, and psychosis is not uncommon in people with bipolar disorder. So we generally discourage people from using cannabis to kind of self-medicate. It can probably do more harm than benefit, but specifically with the cannabidiol, that might be a different story, but we will need to wait for the study results. And I think there are a couple projects looking into it.
Dr. Stern, you, I guess not coming from a biology or psychiatry background, you got to choose, once you understood the technology, where you might apply it. Why the brain? Why did you decide that was the place to go? And I know in your slides you mentioned the hippocampus had a specific desirability for this particular disorder, but what led you to start your work in there, and where do you think we are in terms of our understanding of how different treatments with the brain are going to be used with this new technology?
First, the hippocampus, we are looking now to expand we characterized pretty well, I think, the hippocampal C3 and dentagearus neurons, and we are now trying to expand to other, cortical or fast-spiking interneurons. We are looking to expand to dopaminergic neurons. We want to characterize more areas. We're also building computational models that I wasn't able to show today because of the limits of time, but we're building computational models that take these changes of the electrophysiology that we measure and translate them into currents and potentials in a model. And we want to see how these when we form these networks, how would they behave over time. So these are all things that we're doing. Regarding treatment, this is also something that we also tried, the valproate that Martin talked about, and lithium we're trying in the dish. We also, our results, showing that the fast potassium channels are implicated, or the sodium channels. This gives us, options for possible treatment that will be very targeted towards these specific changes, because there are sodium channel modulators and potassium channel modulators. So this is something that can be developed into specific treatment for the patients.
So Dr. Alda, we're almost at the top of the hour here, so I'm just going to give you one last opportunity to think about that mortality slide that you showed earlier. If we were super successful with Dr. Stern's approach, 10 years out, what would those mortality lines look like? What 10 or 20 years out, what's the possibility and what's the time frame in which we might begin to see the impact of this kind of treatment and approach?
Well, given my age, I think I need to be optimistic. So I would hope that within 10 years, we could see some significant drop. I think in psychiatry, things go hand in hand. I think the research, for instance, also informs the non-medication part of our clinical care. And I've been showing some of the stem cell work to patients who feel that it's very helpful in destigmatizing the illness for themselves and their families. So, there are lots of kind of myths and stigma around bipolar disorder. So even at this stage, it already is helping, and you can take it into psychoeducation sessions with patients. But I would hope that within 10 years we could cut the risk of suicide in bipolar disorder by half, and it would be very substantial because suicide is actually a major leading cause of death, especially in young people. When you compare it with other conditions with high mortality, suicide is really tragic because it affects teenagers, people in their early 20s, and so saving those lives will be very much worthwhile.
Well, thanks so much for quantifying that and giving us really a vision of what this collaboration looks like in the real world and why it's important. I know you were sharing before with Lena your intention to come visit Israel as part of a sabbatical, so that you could actually spend some time directly with Dr. Stern advancing this work that you all have been involved in. We're grateful to you both for this kind of collaboration, and to the Zuckerman Institute, which is making this a priority between great institutions in the United States and North America and in Israel and other institutions around the world. We're grateful to all the scientists and engineers who are involved in this endeavor and really doing something that makes us all proud. That is working on behalf of all humanity. Every time a scientist gets something right, the whole world benefits. And we are so grateful for this focus in this area and for your explaining to us what the significance is. thanks for joining us today on the webinar. We want to invite everyone here today and your friends. Please come join us every Wednesday and Sunday. We're here with our webinar series. This Sunday, we're going to be celebrating another milestone in Israel's history. Just this past month at the Knesset was a commemoration for the 30-year anniversary of Operation Solomon and the Aliyah, or immigration of the Ethiopian community to Israel. We have an amazing panel of a number of people who were involved in those missions, both people who were living in Africa at the time and others who came from Israel and were part of this dramatic rescue. So please come join us on Sunday, join us each week. Everyone, thanks for your time today. Everyone have a safe week.
Thank you.
Thank you for having us.
Thank
Thank you for the invitation.
Thank you. Bye-bye.
Bye.
Bye.